I think we want to get started. We have an exciting talk in front of us. It's a pleasure to introduce the next speaker, Dr. Sergio Grinstein. He's a native of Mexico, but does not come right now from Mexico. He comes from Toronto. He was trained in Mexico City, and then did postdocs, as his wife did, in Toronto and Zurich. Then returned to Toronto, to the Hospital of Sick Children, and rose through the ranks. He's there, has become really one of the preeminent cell physiologists in Canada, and has made major contributions to the field of ion homeostasis, pH homeostasis in cells, but I should say basically he not currently has a Pitblado chair in cell biology at the Hospital for Sick Children, and he's a professor of biochemistry. His recent focus, he has done so many things, it's hard to really enumerate them, but I think one of the field he has recently focused on was innate immunity of cells at a very fundamental level, and that's one of the areas he will talk about today to us on, and the title of his talk is on signaling phagocytosis, receptors, phospholipids, and surface charge. Dr. Weinstein?
Thank you very much, Ulrich. I left Mexico about 30 years ago, so you don't have to worry about swine flu. What I want to talk about today is indeed the innate immune response, and that's illustrated here. What's color-coded here in this scanning EM is a macrophage that's in the process of attracting and interacting with bacteria. And this is a key part of the clearance of infectious microorganisms. But in addition to that, macrophages are also important in the clearance of apoptotic cells. When your cells die, they undergo apoptosis and they don't go to heaven. They actually have to be cleared by macrophages. So macrophages have to open a small mouth to eat bacteria, but also a big mouth to eat dead apoptotic cells. This is a latex bead that's about the size of a apoptotic cell, and you'll see how exquisitely complicated and dynamic this process is. And they can occupy almost half of the cell. They eat two, and then for dessert, why don't we have a third one? So this is a receptor-mediated series of events where the particle, which is coated with ligands, cross-links receptors on the surface of the cell, and this induces a cascade of signals that culminate with the polymerization of actin and the focal delivery of endomembrane vesicles. And together, these two contribute to the extension of pseudopods. When the pseudopods eventually meet at the top of the particle, they fuse and they merge to form two separate membranes. One, a vacuole, that's called the phagosome, and the other one's the plasma membrane that reseals over itself. And a lot had been done in years past looking at the signaling mechanisms by biochemical means. People would take populations of cells, put them in a blender, put them in a gel, run immunoprecipitates or Western blots or whatever, and we learned a great deal about it, but we felt years ago that the reaction is so localized, so transient, and so asynchronous in a population that we would not learn everything that we needed to know unless we looked with more spatial and temporal resolution, and so we went to imaging. And we decided to image phosphoinositides for two reasons. One is that they're key to the actin polymerization cascade. The other one was a purely technical reason. Around that time, it became possible to analyze phosphoinositides using chimeric constructs that consist of the head group of a protein that recognizes the head groups of the phospholipids, in this case, the pH domain of PLC delta, and fuse them with a green or red fluorescent protein to be able to visualize them. So if you take one of these chimeric constructs and you express them in the cytosol of the cell, they'll find their cognate ligand, and the fluorescence will indicate where the lipid is. And so one such experiment is shown here. You can tell me if the lights are good enough to see these things. What I'll show you for the most part are going to be macrophages of a murine cell line in the first part of the talk. And in this case, it's a confocal slice down the middle of the cell. And what you can see is that The construct is partly floating aimlessly in the cytosol, but for the most part, it is bound to the inner aspect of the plasma membrane, which is thought to be the main repository of PIP2. Nothing new about that, but when you look at phagocytosis, you'll notice here pseudopods extending, sealing, and forming a vacuole. Doesn't look very remarkable, except for the fact that the vacuole that just formed originated from the plasma membrane, which is very PIP2 rich, and yet the vacuole is completely depleted of PIP2, and all of that happened in a flash. This is better seen in the sequence of images. The last two steps are magnified here, and you can see that the pseudopods before sealing of a phagosome are actually enriched in PIP2, but as soon as the vacuole forms, there is no PIP2 in the vacuole, but there's lots of PIP2 in the membrane. And the disappearance of PIP2 from the forming phagosome starts actually even before closure of the membrane. You can appreciate this if you use a large particle, like the latex bead here. And you can see that the base of the phagocytic cup is already devoid of PIP2, while the rest of the membrane is very rich in PIP2. So what's going on? What's happening to the PIP2? Well, one possibility is that PIP2 is being converted via PI3 kinase into PIP3. And we have a wealth of constructs now to be able to look at different phosphoinositides. So we can use the pH domain, in this case of AKT, fused to GFP, to look at PIP3. And unlike the other one that binds to PIP2, this one goes and finds PIP3. So when this one is transfected into the macrophages, the picture is totally different. There's virtually no PIP3 in the cells at rest. The construct is cytosolic and doesn't know where to go. But as soon as phagocytosis starts, you can see an inkling of what's going to happen here, but this is a movie. You'll see the appearance of PIP3 in multiple phagosomes. And the movie is pretty self-explanatory. Every time you have a phagosome forming, you have very localized formation of PIP3. You can see a phagosome here that hasn't yet closed, but has a tremendous amount of PIP3 formed exclusively at the base of the phagocytic cup without extending to the bulk membrane. When the phagosome seals, when there's no PIP2 left, you have lots of PIP3. And then a minute later, it starts vanishing and disappears within 2 minutes. If you try to do this biochemically by HPLC with tritiated inositol, you're up the creek without a paddle. So don't try that. Now, PIP2 is partly going to PIP3, but we believe the bulk of the PIP2 is actually being degraded by phospholipase C to form diacylglycerol. And we believe that because, yet again, we can use diacylglycerol-specific constructs, in this case, the C1 domain of PKC delta. And to make the images technicolor, we can use a yellow fluorescent protein and put a blue fluorescent or cyan fluorescent protein in the pH domain that can see PIP2. And you get these types of images where prior to phagocytosis, the membrane has lots of PIP2, but no diacylglycerol. Even when the phagosome is beginning to form and the pseudopods are extending, which is magnified in the inset, lots of PIP2, no diacylglycerol. But if you look a minute later, the part of the phagosome that's in closest contact with the particle is beginning to lose PIP2. And right there and then, you see the appearance of diacylglycerol. And then a minute later, when the phagosome seals, the phagosomal membrane is totally devoid of PIP2. But that's where the diacylglycerol is present. And a minute later, the diacylglycerol fades away. So again, very difficult to monitor diacylglycerol appearance and disappearance by biochemical means. So all of that was published years ago, and we knew what was becoming of the PIP2, but then we started wondering, what is the implication of losing PIP2, and why do we worry about PIP2 disappearance? Not only is PIP2 important itself for actin polymerization, but PIP2 is a tetravalent anion. It's all localized in the inner aspect of the plasma membrane, and it's supposed to represent between 2 and 5% mole percent of the lipid. So that means there's an awful lot of very negatively charged lipid. In addition to that, there is PIP and phosphoserine, which makes this membrane uniquely negatively charged. And as you know, a negative surface will bind and attract ions also because of the surface potential. So what are the implications of altering the concentration of PIP2 in terms of the surface charge, and what are the consequences in terms of protein binding? Stuart McLaughlin has made a living out of looking at polycationic proteins that bind to polyanionic surfaces like the plasma membrane. So is this happening and is this changing during the course of phagocytosis? So what we wanted is to measure the surface charge of the inner aspect of the plasma membrane during the course of phagocytosis in intact cells. And this is surface charge, not Nernstian voltage. This is not something you can measure with patch clamping or intracellular electrodes. This had to be measured differently. And we looked in the literature how one measures surface charge in internal membranes, and we came up with a blank. And so we had to design our own approaches, and I'll spare you the trials and tribulations. I'll give you the punchline. We have to measure surface charge, not surface, not charge. So the probes are coincidence detectors that have a surface targeting motif and a charge identifying motif. So they have a hydrophobic component, in this case farnesylation, myristylation, or hydrophobic side chains of amino acids. And they have a polycationic stretch of 8 residues that can be lysines or preferably arginines, so we don't deal with ubiquitination issues. And to know where the probes are, we put a fluorescent protein at the end. So these are all encoded in plasmids. We transfect them into cells, and we ask, where do they go? And whether they're myristylated, amphiphilic, or prenylated, they go almost exclusively to the inner aspect of the plasma membrane. And we believe that's the case because the plasma membrane is the most negatively charged surface of the cell because of the enrichment in phosphoinositides. How do we know that this is really charged and not something else? Well, if you mutate the 8 charges down to 5, instead of all being in the plasma membrane, the construct is now partly in the plasma membrane, but some of it is now in endomembranes. If you go to +2, you virtually see it in every membrane and very little in the plasma membrane. There's a very good linear correlation between the amount in the plasma membrane and the number of cationic charges. So we feel we have a probe to measure surface charge in intact cells, but is this a dynamic probe or is this just stuck there? Well, the way to tell is if we manipulate the surface charge in a manner that is predictable. And we can do that by adding calcium and ionomycin. So calcium gets into the cells and does 3 different things. It directly shields the charge. It activates phospholipase C, which hydrolyzes the head groups of PIP2. And it also activates the scramblases that will scramble phosphatidylserine to the outer monolayer. So all 3 effects combined should produce a large drop in the surface charge within minutes. And if that's the case, the probe, if it's a dynamic probe, should come off the membrane. Now, those of you who have added calcium and ionophore to cells, you know, if you leave them long enough, they turn into snot or yogurt or something. So you want to make sure your cells are alive and well and intact. And to monitor for intactness and well-being, we transfect the cells with plasma membrane markers that are hydrophobically attached. So they're not sensitive to the surface charge. So either by GPI linkage, by a transmembrane domain, or by acylation chains. And to these probes, we attached a green fluorescent protein. They look like this. They're at the plasma membrane, but also in some endomembrane compartments. But now we're in a position to doubly transfect the generic garden variety membrane marker with a surface charge marker. And initially, they look pretty much the same. But we can ask now the question, what happens if we let calcium in? So this is a movie. This is the time course after addition of the calcium ionophore. Initially, not much happens, but when calcium builds up sufficiently, the surface charge changes drastically and the probe comes off the membrane. And the same thing happens whether you're using amphiphilic, prenylated, or myristylated, except some of them don't enter the nucleus and some do. And all the garden variety markers tell you that the cell remains intact. You get the same type of drop in the surface charge when you just deplete ATP in the cells by inhibiting glycolysis and mitochondrial metabolism, because the phosphorylation of phosphoinositides and the flipping of phosphatidylserine require ATP. So after about 30, 40 minutes of ATP depletion, the cells look like this. Or you can do the same thing using local anesthetics like dibucane, which are bivalent cations. They insert into the membrane and change the surface charge. And all of them drastically reduce the partitioning of the surface charge probe. So if you haven't forgotten where we wanted to go, the idea was to find out what happens to surface charge when the composition of the phagosome changes during phagocytosis. So here is a dual-labeled experiment where we have surface charge in red and the garden variety marker of the membrane in green. And this is important because I mentioned before that there's delivery of endomembranes to the surface. So the membrane tends to get diluted a little bit during the course of phagocytosis. So we had to normalize for that. So here's a course of phagocytosis. You can see phagosomes already formed, some new ones forming. You can see the membrane leaves residues of the plasma membrane there. They're very clearly observable, but the surface charge has dropped to the point that you can't see it. And here's another case where you have 5 phagosomes that just formed, very clearly retaining remnants of the plasma membrane, but with a very drastic change in the surface charge. So that's nice. The surface charge is indeed changing as one would have anticipated, but so what? What are the consequences of this? And I mentioned that polycationic proteins tend to stick to the membrane electrostatically. So is that changing? And a beautiful experimental paradigm is a comparison between KRAS and HRAS. These two proteins are very homologous in their GTPase site, in their effector binding domain, but they have a C-terminus that's called a hypervariable region that differs drastically between them. KRAS has a farnesylation site and a polycationic stretch, just like our probes. HRAS has a farnesylation site, but two cysteines that are palmitylation motifs. This guy inserts into the membrane hydrophobically and should be insensitive to the surface charge. This guy attaches to the membrane electrostatically and should respond to changes in surface charge. Now, let me show you what happens when you transfect the full-length proteins. This is KRAS, this is HRAS. Both of them go to the plasma membrane, but by different mechanisms, and I'll show you that that's the case. KRAS is almost entirely in the plasma membrane electrostatically because of these charges, and we know that because if you mutagenize the tail to make it go from 8+ down to 5+, it actually looks very much like our little 5+ probes. Moreover, if you take the full-length 8+ KRAS and add ionophore, it comes off the membrane, just like the probes did. Now, this is HRAS, which is attached hydrophobically. If you now add calcium and ionophore, nothing happens. It stays at the membrane because the attachment site is hydrophobic and insensitive to changes in surface charge. So here's a comparison of KRAS and HRAS. KRAS in red, HRAS in green, both of them full-length wild-type proteins. Both of them go to the plasma membrane. The membrane's yellow. Then we allow phagocytosis. And this is after phagocytosis. It's very obvious that HRAS, the hydrophobically attached one, remains associated with the phagosomes, whereas KRAS has fallen off. So here's a means of separating, physically diverging between the signaling of HRAS and KRAS, which are very homologous proteins, on the basis of a surface charge change of the target membrane. Now, we're not quite sure what KRAS does in phagocytosis, but RAC is the driver of actin polymerization in phagocytosis. And like RAS, RAC has farnesylation and a polycationic stretch. And I'll make a long story short. To get around problems of GEFs and GAPs and so on, I'm not going to give you a headache, we used constitutively active RAC, the Q61 form, which is with a red fluorescent protein. And we co-transfected it with one of those generic membrane markers. Both of them go to the plasma membrane. And we know that RAC1 goes to the plasma membrane electrostatically because it is sensitive to calcium and ionophore, etc. Following phagocytosis, the green membrane marker remains. The red RAC falls off the membrane. This is not because you activated a gap. This is not because you terminated a GEF, because this is constitutively GTP bound. You eliminated RAC from the phagosome because the surface charge changed. So here's a manner of terminating signaling independent of regulation of GEF or GAP. Okay, so what I wanted to do today is give you two vignettes of the work that we do in the lab. This one that relates to surface charge, where I told you that the membrane is very negatively charged and that the phagosome loses charge, and for that reason alters the targeting of proteins. And this work was done mostly by a couple of students, Tony Young and Rob Botelho, and postdoctoral fellows and colleagues, with collaboration from a number of people that were invaluable in doing these studies. The second vignette was done with a second group of collaborators from the Scripps Clinic, Kulut, Jackaman, and Gaudens-Danusser. It also deals with phagocytosis, but with one of the earlier stages of the process. So I mentioned that this is a receptor-mediated event, that a multivalent particle comes and cross-links ligands, and that triggers signaling. And we've assumed for years that the receptors exist as monomers, that unliganded receptors are homogeneous in the surface of the membrane, and that they move randomly in Brownian fashion. And so that when the target comes, they just wiggle around and go and bind and cross-link. And that applies very well when you drop one of these bowling balls on top of the cell, because you have lots of time. But if it's a bacterium that comes by and fleetingly makes contact with the cell, do you actually have enough time to cross-link the receptors? And you can do some back-of-the-envelope calculations, and you find out that maybe not. And so we decided to reanalyze all of these premises using this phagocytic receptor, CD36, which is not only a phagocytic receptor for apoptotic cells or for malaria-infected red cells, it's actually a phagocytic receptor for oxidized LDL. So it's the one that gives you atherosclerosis, plaque, and heart attacks when you go to McDonald's one time too many. And what we wanted to do is measure primary cells and endogenous receptors. And so we used antibodies and labeled antibodies, but we cannot use full bivalent antibodies because we get cross-linking. So all the work that I'll tell you here on after are Fab fragments, primary Fab that we made in the lab, and the secondary Cy3 labeled Fab that we buy from Jackson. So now we can measure individual receptors in intact primary cells. So these are human monocyte-derived macrophages now. And the first question we asked is, are the receptors really monomers homogeneously dispersed? Fix the cells and labeled with low doses of primary, constant amount of secondary. When you measure the appearance, you get single spots of about homogeneous intensity that you can plot in this histogram. As you continue to increase the concentration of the primary at constant secondary, you start getting brighter and brighter spots. Not only more spots, but brighter spots. And these spots are actually multimers of the monomeric spot. That tells us that at least in fixed cells, this thing is in multimers. And we can validate that if we photobleach by just exposing to light one of these bright spots. You go from here to here to here to here, and actually the units of bleaching are comparable to the intensity of the least intense spots, the monomers. So in fixed cells, at least, the receptors exist as multimers. Now, this could be an artifact of fixation. How about in live cells? So we're now able to measure with electron-multiplied cameras single spots and find their behavior in live cells. So this is a little movie of one of these spots. It's a nice, bright little spot, but before you know, it splits into two. And if you keep looking, it splits into three. It comes and goes, and they're dancing around, and you can see it better in this gallery of images. You start with a bright spot, breaks into two, goes to three, collapses into two, goes back to one, etc. So, whoops. These are multimers, and these multimers are metastable. They're forming and unforming. And this is not a single event. This happens throughout the cell. So what you're looking at here is epifluorescence of a cell. This is the body of the cell. This is the lamellipodium. And you can see that in that area that I'm highlighting, but elsewhere too. These particles are dancing around, and they kiss, and they form multimers, and they separate, and they go away. And for the longest time, we were really happy looking at these images like the village idiot, until we figured we have to do some quantitation of this. We didn't know how to, and that's when we contacted Gaudens and Kulut at Scripps. And they made and had fantastic software that they've been improving for us to track these particles, and the little yellow lines here track the individual particles. And what becomes very obvious when you look at the tracking is that these are not random moving particles. These guys are moving, at least in part, in a rather linear fashion. And again, Gauden's devised software to quantify arbitrarily, if you want, what we call linear. So we define parameters that are arbitrary to decide what's linear and what is nonlinear, and it's color-coded in blue and red. So there's about 30 to 40% of the particles that are clearly moving in a linear radial fashion. So the obvious suspicion is this is the cytoskeleton that's doing this, and is it actin or microtubules? And because they're primary cells and we cannot transfect them very well, we can use only pharmacology, but the pharmacology is reasonably clean. You treat the cells with latronculin, and compared to the unperturbed cells, which have a lot of linear tracks, you get very few and very short linear tracks. So actin is required for this linear motion. So what we expected to see if we stained these cells for actin were nice linear tracks, microfilaments, or something where the particles are moving, but when you stain these guys for actin, it doesn't look anything like linear tracks. It looks like what Amira showed before, a meshwork of submembranous actin that's pretty disorganized. So how do you go from this to linear motion? In fact, what is linear and what is radial in these cells are microtubules, which are stained here. And if you stain CD36 as well as microtubules, you'll see that very often CD36 seems to be lined up on the microtubules. And if you do statistical calculations, you'll see that this is not a random occurrence. These guys tend to line up preferentially on the microtubules. So are microtubules necessary for this motion? We rupture them with nocodazole, and yes, lo and behold, you can break the linear translation with nocodazole as well. So what I've told you so far is a fraction of the CD36 moves linearly, and that the linear motion depends on the cytoskeleton. So the obvious suspicion is that there are motors that are driving the particles back and forth along these microtubules or microfilaments. We don't have to speculate. There are actually very well-defined criteria to decide if something is motor-driven or if it's random motion. For one thing, motor-driven motion is anisotropic, meaning it is not random. It's directed, and we've seen that it is directed already. It should have a constant runtime because motors have a defined catalytic cycle. And the speed should be usually different to and away from the nucleus because you have kinesin this way, dynein the other way, or myosin one way, kinesin the other way. So you expect different speed in both directions. In contrast, if it's random motion, it should be isotropic. The runtime should be 2 to the minus n, and I'll explain that in a second. And the speed should be the same in both directions. So why don't we analyze each one of these criteria independently? Let's start with the speed, which is the weakest of the criteria. If it's a motor, you expect a very well-defined speed maximum. Every time it hitches onto a kinesin or a myosin or whatever, you should get a preferred speed. So we measure the speed in both directions. And what we find is instead of finding a very sharply defined speed, we find a broad shoulder of multiple speeds. And more remarkably, the speed to and away from the nucleus is identical. Now that was disturbing because that is not consistent with motor-driven motion. That is consistent with random motion. So, well, maybe something's wrong with our measurements. Let's look at the runtime and see if it is constant or if it is according to random motion. So if you have a motor, let's say the stroke of your motor is 150 milliseconds in duration. It goes from here to here in 150 milliseconds every time. So if you're acquiring with your camera frames every 50 milliseconds, then you'll catch each stroke of the motor in 3 frames. So 50 milliseconds, 150, 3 frames. If you're actually acquiring faster, if you're acquiring every 25 milliseconds, you'll have 6 frames moving in the same direction before the motor completes a cycle. Conversely, so in this case, the runtime in number of frames varies with the acquisition rate. Conversely, if you're looking at random motion, if you have a particle and that particle sitting here, it has a 50-50 chance of going to the right or to the left. It's random. It's 50%. Now this particle here now has a 50-50 chance of going to the right or to the left. So the probability of the particle going 2 frames in sequence to the right is going to be 0.25. In fact, the probability of the same particle going 3 frames to the right in sequence is going to be 0.125. So for random motion, the runtime should be proportional to 2 to the minus n. And it should be independent of the acquisition rate, because there's no defined specific motion. This is going to be random. So let's look at what happens. This is the runtime in the number of frames, and this is the frequency. So how many particles change direction after 1 frame? That's about 50% of them. How many particles change direction after 2 frames? That's about half as many as 1 frame. How many after 3 frames? Well, half as many as before. This is consistent with random motion. Now, what happens if we do this at different frame rates? If you change from 10 hertz acquisition to 33 to 125 hertz acquisition, you get exactly the same behavior. So the runtime seems to be conforming to 2 to the minus n, and it's independent of the frame rate of acquisition. This is typical of random motion. So this is crazy, because we have this indicating random motion, this indicating random motion, and yet I told you right from the outset that we're looking at something that's moving linearly. So by definition, it's anisotropic. Well, let's maybe look at it in more detail. To look in more detail, we want to measure the displacement in the long axis, in the linear axis, and compare it to the displacement in the perpendicular direction. We need very high resolution. So how do we accomplish this? Well, we go from Psi3 labeled FRAP to quantum dots. Why do we use quantum dots? Because they're brighter, and we can determine their position better. Why is that the case if the quantum dot is actually bigger than Psi3? Well, both of them are actually limited by the diffraction limit of the microscope. We're not seeing the fluorophore, we're not seeing the quantum dot, we're seeing a property of the microscope which is really dictated by the wavelength and the numerical aperture. And this is much, much wider than the size of the fluorophore. However, the distribution of light is Gaussian. And if you measure accurately the distribution of fluorescence in the different pixels, you can calculate mathematically for each one of these dots at any one time the Gaussian distribution of light. And you ask the computer to calculate the median of the Gaussian. The median of the Gaussian is the precise position of the fluorophore, which you're determining with sub-diffraction resolution. So you're actually defeating the diffraction limit of the microscope if your measurements and calculations are accurate enough. So we can now take quantum dots and make movies and track these quantum dots more precisely. So here's a movie, and you'll see these guys wiggle. There they go. This guy's going forward. Now it goes backward a little. This guy is going forward. Now it goes backward. This one's going backward. And you can see they are moving linearly, but if you look in detail, they're actually wiggling a little bit. And if you blow up one of these images and look at it in detail, you'll see what exactly the particles are doing. Now, the color coding here is just the time course of acquisition. This is where we start. This is where we finish. If you look at the particle here, and you don't look at the particle again until the particle has reached here, that looks pretty darn linear. Right? But if you look at the particle at a much shorter time interval, the particle is doing all kinds of this crazy moving around. That doesn't look linear. In fact, the closest analogy to this is a drunken sailor in a dark alleyway. Doesn't know where it wants to go, it's bouncing off the walls, and if you wait long enough, he'll make it out of the alleyway. But if you sample where the sailor is going over short intervals, it is pretty random. Here's a comparison of the perpendicular and linear rates of motion at different sampling intervals. If you sample at long intervals, then you will see that the linear displacement is much greater than the perpendicular displacement. That's because you allowed the sailor enough time to find the way out of the alleyway. Right? We can do this both with Cy3 and with quantum dots because it's long acquisitions. But if you want to make very short acquisitions, which you cannot do with Cy3, you use the quantum dots, as the acquisition time gets shorter and shorter, the anisotropy is lost. The perpendicular motion becomes pretty darn close to the longitudinal motion. So this is not truly, in the short term, anisotropic. This is practically isotropic if you sample with a high enough frame rate. So if you take that into account, it's nearly isotropic, has a runtime of 2 to the minus n, and the speed is the same to and away from the nucleus. So you're really dealing with random motion. But if it's random motion, why is it going in the long run in the linear direction? So what we think is happening is that this is random motion within a confined space. And we believe the confined space is given by a breakdown of the actin meshwork in some fashion induced by the microtubules, whether the microtubules are breaking right through the actin mesh or maybe pulling the actin mesh a little bit away from the membrane, allowing a trough, a channel, where there's no actin barriers and the particles can now move freely back and forth. So who cares, right? Why is this important? Well, it's important for the considerations that I mentioned early. If you have 2 receptors or 3 receptors or 5 receptors that have to come together over a short period of time when the particle is in the neighborhood, if you have too many degrees of freedom, if you have these guys in 3 dimensions, they'll never find each other. If you have them in 2 dimensions, they might, but they might not. If you have them all confined in 1 dimension, so you're diminishing dimensionality, there's a much better chance that those receptors will come together in a short period of time. In other words, it's much easier to rope in the horses if they're inside the corral than if they're running out in the fields. So if you have 3 or 4 or 5 receptors bouncing up and down this one little narrow alleyway, and a particle happens to come by, your chances of that particle capturing 3 receptors in a meaningfully productive physiological way are much greater when you've reduced dimensionality. And again, this work was done in collaboration with Collud and Gaudens at Scripps, and the work in my lab was done by 2 postdocs, Hirotaka Kuwata, Nicholas Tourette, in collaboration with my colleague William Trimble. Well, thank you.
Well, thank you very much, Sergio. Are there questions for Sergio? Can we get the microphone to the... No, I didn't explain it because we really don't know the actual molecular mechanism. We know that this correlates with the directionality of microtubules. We know it depends on microtubules, so we assume, and we have no molecular mechanism at all, we assume that somehow in those areas where microtubules run near the plasma membrane, that there is either a breakdown or a detachment of the actin meshwork that enables this to happen. Don't ask me about molecular mechanism. Do microtubules actually come close to the membrane? Because most people picture them as running right in the middle of the cell. If you take these cells and you look with turf, you have lots of microtubules within the turf illumination field, which is 150 nanometers. So they do come very close. That's about all I can tell you.
Yeah, we've tried to do this with John Hardwick and with John Heuser, but there have been problems. And not technical problems, but other problems.
There's another question. Here, we need to know.
So, what do you think of the huge accumulation of PhD? What's that for?
What's that for? Well, on the one hand, you can rationalize it as terminate or making the PIP2 disappear, but certainly diacylglycerol itself will be activating anything that has a C1 domain, protein kinase C or some of the exchange factors. So we know it activates protein kinase C epsilon and delta, that's been reported. And we also know that it activates an exchange factor for RAS and RAP1. And we know that activation of RAP1 will couple one receptor with another, and there may be other things.
So does that have anything to do with the endocytic process?
With phagocytosis, yes. If you prevent the hydrolysis of PIP2, you prevent phagocytosis. If you inhibit diacylglycerol activity by using analogs, you inhibit phagocytosis.
So that product, let's say the PKCs, for example, you're activating the PAG, what is their purpose?
Yeah, there's a lot of debate. Some people believe they're essential. Some people don't believe that at all. I think part of this, we haven't looked at it ourselves, but I think part of the discrepancy comes from the fact that there are multiple types of phagocytosis induced by multiple receptors, and multiple cell systems have been used. So, an FC receptor and a complement receptor and a Mannan receptor signal through different pathways. Some may require PKC, some may not, and most of the conclusions are from pharmacology, which is not necessarily PKC targeted.
Another question?
So have you screened for any other proteins that have the magic octa-lysine for this surface charge interaction? Or is it you look to see other proteins that would have that?
Right, yeah. No, we haven't done this systematically, but there's a beautiful paper by Tobias Meyer where he took the old 63 or 66 RABs, and he looked informatically at which ones of the RABs have polycationic tails and how does that correlate to the partition in the membrane. There's a beautiful relationship between the polycationic ones, the ones that like to go to the plasma membrane. Yeah, now we have looked at some other polycationic proteins, and they don't have to have polycationic linear stretches. If they have an electrostatic vector, because they fold three-dimensionally in a manner that exposes a cationic patch, they'll also do it. And there are a number of proteins. So the kinase that makes PIP2, BIP kinase 1, has a beautiful patch, binds to the membrane, and the binding is electrostatic.
Well, I think we have to go on. Thank you very much. Dr. Schilling will give the next one.
So I'd like to introduce our next speaker, Dr. David Clapham. David got his undergraduate degree from Georgia Tech in electrical engineering. He then moved to Emory into the MSTP program, where he got his medical degree and his Ph.D. in cell biology and anatomy. After several years of residency at the Brigham and Women's Hospital and Harvard Medical Center, he then went for a couple of years to Germany to work in the lab of Erwin Nair at the Max Planck Institute. In 1996, or just before that, he took a position in the Mayo Clinic in Rochester, Minnesota, as an assistant professor. He then moved up the ranks very quickly and became a full professor in 1993 at the Mayo Clinic in the Department of Pharmacology and Physiology and Biophysics. In 1996, he left the Mayo Clinic and moved back to Boston, where he's currently a professor of neurobiology, professor of pediatrics. He's also the director of cardiovascular research at Children's Hospital in Boston, and he's the Aldo R. Castaneda Professor of Cardiovascular Research, and he's been an investigator of the Howard Hughes Medical Institute for the last 12 years. His research initially focused on the interactions between G-protein-coupled receptors, G-proteins, and ion channels, and the mechanisms by which the ion channels are regulated by G-protein subunits. He quickly expanded into areas of calcium signal transduction and the mechanisms by which cells increase their calcium. And this, of course, brought him most recently into the area of trip channels. And he's made important contributions to our understanding of all of these individual areas. And so without further ado, his talk today is entitled Novel Functions of Ion Channels. And I think he's going to focus today on magnesium. David.
Okay, I'm going to tell you about, we work on a lot of different ion channels. I'm going to talk, though, mainly about trip channels today. And what I want to tell you about is channels that I think have a lot to do with their own incorporation into the membrane. Trip channels, in many cases, seem to govern their own availability in the membrane and therefore have some function in vesicles. And so I'm going to try to convince you that that's one of the things that they do. So, as you all know, the only real point of ion channels is to regulate calcium. That's what they're for. And if you can think of a counterexample, let me know. Because the point of all the timing of action potentials and the delays and the links is to regulate the timing and entry of calcium, because it is calcium that initiates contraction, secretion, and all of the myriad of events that we know. And calcium is the reason it's so important is that it's a huge gradient from outside to inside, a 20,000-fold gradient, and that makes it what that double charge and a huge gradient is what makes calcium such a special signaling agent. Ion channels conduct calcium and other ions, of course, and they are very much like transistors. This bottom is from Fred Sigworth, a friend of mine at Yale, who compared the current voltage relationships of a transistor and an ion channel, and you can see that they're very similar. In fact, ion channels can be steeper than transistors in their current voltage relationship. Their job are switches. They are switches to regulate calcium. So, what kind of architecture do they have? Well, they're all stem from this kind of architecture. They have probably the simplest ion channels have two transmembrane domains, and they often have a kink in this second transmembrane domain, and often things pull on this end or this end to open this channel. G-protein gated inwardly rectifying K channels, and many inwardly rectifying K channels are of this class, just a pore. They tetramerize to form that pore. And then there's also one channel that we cloned a couple years ago. It's really just a voltage sensing domain, and it is a channel only by virtue of the fact that the voltage sensor domain allows for water to sneak into the middle of this. And once in a while, there's a little water channel that forms that allows protons to conduct through it. It's perfectly selective for protons. It's a voltage-gated proton channel. But this S1 through S4 region and this pore domain got joined somehow in evolution and became the standard for most of these voltage-gated ion channels. And of these, there are about 150 types in mammalian systems where this is called the voltage sensor domain or the voltage sensing ligand domain. Many things bind to it, but its job is to make a motion that results in the movement of this gate here between S5 and S6 to open this channel. So the channels that we work on, about half the lab works on trip channels. We also have been working a lot on mitochondrial calcium transporters, that is channels that are inside mitochondria intermembrane, trying to find out what they are, this voltage-gated proton channel, and we've been working a lot on ion channels of sperm lately. So, but I'm going to focus on trips. What defines a trip? The trips have not such terrific homology between themselves. And I was involved in the nomenclature originally, and just to prove that they didn't have, the trips is a little bit misleading to call them all trips, is that the people in the mucolipin field and the polycystins field, we didn't include them originally in the nomenclature, and they called us up and said, well, why aren't we trips? And we said, well, why not? And that's how, that's how they got in there anyway. But there's not that great of a degree of homology between them. The TRP channels are generally channels that are non-selective. They bring sodium and calcium into cells. 2 of them are calcium selective, fairly calcium selective, and a couple are sodium selective. But most have reversal potentials around 0 millivolts and let many cations through. They're slow, tend to be slow. They're weakly voltage sensitive. They have intrinsic ligands like PIP2, but this is true of most ion channels. But also nature has evolved compounds that activate them. And I assume that's because they're often on sensory organs. So capsaicin, hot pepper, menthol, for example, are plant mechanisms to either repel or attract animals to help them in their survival. And often they are linked by GQ-linked receptors to phospholipase C. This is one of the most common characteristics between them. So I mentioned some of these in the last slide, but this is, to me, is a central problem of the TRP channel family, is what gates them. It isn't capsaicin. They didn't evolve to be gated by capsaicin or menthol. They didn't evolve to be gated by any of the things that have been tested so far, perhaps pH in some cases. But many of the things that you've read about or heard about in the activation of TRPs aren't very strong activators. Fatty acids, arachidonic acids, mechanical stretch is incredibly weak activator of TRPs. Osmolar sensing, very weak. Nitric oxide is weak. So even though these get a lot of attention, these various TRP, I would say they're all modulators. I'd say we still don't know, in most cases, what gates TRPs. And that's the main thing I would like to know, is if there is some common gating mechanism. Okay, so where did the first TRP appear? It was invented in yeast. Yeast have a TRP channel, and it's in the vacuole. This vacuole is sort of like a capacitor that allows yeast to live in different degrees of osmolarity, like distilled water. They could adapt by putting the excess water in their vacuole and letting it out when they're in more hypertonic water and so forth. This channel has never been measured because it's in this vacuole, but it is a TRP channel and presumably non-selective. So when we started looking at using turf to look at what I thought TRPs were going to do at the time, which was I thought they were going to have big effects on cytoskeleton. They didn't, but instead we found that they were commonly in vesicles. And one of the most striking cases of TRPs in vesicles was TRPC5. We found that TRPC5 was in these very characteristically large vesicles, and when 318 nanometers. So this is much larger than a synaptic vesicle, which is about 40 nanometers. This is a turf image showing that this vesicle kind of randomly moves around, and that's shown here, until it reaches some attachment point, a microtubule or something, which brings it right up to the surface. And so TRP, this is loaded with C5. It turns out that C5 vesicles get loaded onto Stathman and nerves and go out to growth cones where they sit under the surface. And within a couple of minutes after growth factor exposure, they get put into the membrane where their channels act to bring calcium in, probably for arrest of growth cone movement or steering of growth cones. And so they regulate their own incorporation, but also affect biology through their ion channel function on the cell surface. So why would they be involved in fusion with the membrane, and why would they be then incorporating into the membrane? Well, vesicles, of course, have a whole snare apparatus, and that snare apparatus is present on all kinds of fusions, not just between vesicle and membrane, but between vesicles. It almost always includes synaptotagmin, which is a calcium-dependent zipper, so to speak. It is an initiator of the zipper, I guess you could say, that's calcium dependent. And so trips may be in vesicles to allow conduction out of the vesicle of calcium. It may be that trips on the surface can let a little bit of calcium in to initiate fusion as well. But these are the ones that are fairly certain to participate in vesicle formation: TRPC5, TRPM7, TRPML3, mucolipin-3. And these are ones that haven't been really proven, but I suspect are also doing similar jobs. Not all trips necessarily, but a good fraction of them. And then some trips, like the mucolipins, mucolipidosis is one of the mucolipidoses, is a disease that's caused by a mutation of TRPML1, and that disease is a failure in endocytic and lysosomal sorting. And the reason we think that happens is that mutants of TRPL1 cause defects in intravesicular fusion events. That is, TRP channels are in this membrane and they may release calcium, initiating a snare apparatus to induce fusion with another vesicle. Okay, how would they pass current? Well, why do we think this? I was always puzzled by the IV of TRPs. They have a great outward rectification, so they look a lot like potassium channels in this regard, except that their outward current is even more shifted than potassium currents generally are. This is in the non-physiologic range. Most of what current they pass is out here in a non-physiologic range. This is the physiologic range, and for many TRPs, their currents are very small in the inward direction compared to the outward direction. But if you were in a vesicle, you would flip that IV, and the current would go from inside to outside and would get increased depending on this voltage. Now, the questions that are not known, things that are the driving forces. So certainly there's a calcium driving force from inside a vesicle to out, but the voltage, the transvesicular voltage, is very poorly understood. One of the other things that makes me think about TRPs being in vesicles is that they are many of them are strongly potentiated by pH. And as you know, many vesicles, because of VATPases, have pHs on the order of 3 to 5. Endosomes and lysosomes certainly do. And so, and also that they're potentiated by PIP2, which is not in vesicles, but is where this part of the ion channel binds, it is in plasma membranes. So PIP2, which would be a marker for the plasma membrane, would activate the intracellular part of a TRP channel. All of those things make you wonder if one of their main jobs isn't in vesicles. But I'm going to tell you about a channel that doesn't work in vesicles but is also on the plasma membrane. And I'm going to focus primarily on its job in the plasma membrane. We call this TRPM7. It's had a lot of other names. It used to be called TRPLIC. We originally discovered it a long time ago in a yeast 2 hybrid assay. And it, to me, is the most mysterious, but probably also the most important TRP, I think. And I'll tell you why. You'll see why in a minute. So M7, it has a close sister, by the way, called TRPM6. It's about 80% identical, but M6 is mainly in kidney. M7 is ubiquitous. M7 is unusual. It has 1,800 amino acids. This is as large as a voltage-gated sodium channel, all 4 domains. This is one of a part of the tetramer. These form tetramers as well. So they have this huge amino and carboxy terminal regions. This last part is a kinase, and this kinase was crystallized almost immediately after M7 was found, crystallized by John Currien's lab, and found that it folded, even though it was of a group called the ATP grasp domain proteins, of which there are only a small number of kinases. It folds like a protein kinase A. Here's the unusual thing: it has a zinc in it, which is a high affinity and probably just helps it fold the way it is. But its active site is very similar to protein kinase A. This is a dimer of this M7 kinase. We call it PLIC, or we all call it the M7 kinase. The reason we call it PLIC is that this kinase was originally found. We were looking for, and we knew that trips commonly were regulated by phospholipase Cs. We thought we'd look in yeast 2 hybrid assays with phospholipase C and see what we could pull out, and we found TRPM7. TRPM7 interacts with PLC beta 1 and 2, for example, and 3 and gamma 1, and it interacts through the kinase domain. So it also is a functional kinase. That is, it doesn't just look like a kinase. It will also phosphorylate things. What it normally phosphorylates is one of the main questions we have, but it will phosphorylate myelin basic protein, common substrates where you check for things for phosphorylation. It will phosphorylate annexin, for example. That doesn't mean that's what its job is in vivo, is to phosphorylate those, but it is a kinase, and it phosphorylates itself. Okay, so back to the channel part of this protein. The inward conductance of IM7 is especially small. Here's the IV relation. See, there's plenty of current when you get out to plus 60. It's non-selective, but here's another odd thing. It is permeant to magnesium and calcium. No self-respecting ion channel does this. So the reason is that magnesium just does not give up its waters. Magnesium tightly binds 6 or 8 waters around itself. Calcium can be, we assume, dehydrated like potassium in a selectivity filter, but magnesium, the energies are too high. So this means that it has a big hole in it, and that the conductance going in is not that high, meaning that there's probably some binding of these charges to the external mouth. Intracellular magnesium blocks the channel. It's called the MIC channel, or magnesium inhibitable channel. It's activated by PIP2. It's greatly potentiated by phospholipase C, and low pH outside, like I was mentioning in the exocytosis part, activates this channel. This is to show you that PIP2 can activate the channel, although I think I would rather call it potentiation. Here's carbachol activating M1 receptor, and we're applying carbachol here and then washing it out and applying it again and so forth. And you can see that when you apply carbachol, the current changes dramatically. So what happens here is that carbachol causes hydrolysis of PIP2, phospholipase C, and just as Sergio was talking about, phospholipase C hydrolyzes PIP2. In most cases, as I told you, phospholipase C potentiates TRP channels. In this case, phospholipase C is degrading PIP2 and causing this channel to turn off. And this shows a PIP2 sensor, which was a pH delta 1 domain tied to GFP, and showing that the time course of the PIP2 change and the current change, current over here, and this is normalized TRPLIC in red, and this is the translocation of or hydrolysis of PIP2. They're exactly the same time course. Okay, so here we have a channel that's activated by PIP2, turned off by GQ-linked receptors in some cases, phospholipase C, that is ubiquitous everywhere, and we don't know what gates it normally. So one of the odd things about this channel is that you can overexpress it and break into a cell and see the current immediately, but in most cells what happens is that you break into a cell and the current develops over time. It takes minutes to develop, but it just gets bigger and bigger over time. It is a common source of errors by electrophysiologists who aren't aware of it and don't use magnesium inside their pipettes, because if you don't block with magnesium, this current develops over time and is commonly mistaken for other things. So it's good to, if you're starting to look at TRPs, to be aware of TRPM7 and watch and use high magnesium, like 5 millimolar magnesium, to prevent its activation. So something is diffusing out of the cell when we do this to help activate it or turn it on. Then we got kind of stuck. What else to do with TRPM7, ubiquitous? So we knocked it out. It's a lot of trouble with knocking it out. There's lots. It has poison DNA. Every time you try to make a mini prep, it gets mutations. You have to do a lot of things to get to work with TRPM7. And we knocked it out in 4 different ways. And the 4 ways that we used all gave out the same result. Why did we do it 4 different ways? Well, one is that we wanted, this is a GATA1 CRE, that's a global knockout. This is a global knockout. We wanted to make sure people had maintained that the point of M7, and you probably saw it for M6 on one of Rick Lifton's slides, this was a common idea that the point of these channels is to bring magnesium into cells for magnesium homeostasis. And I'm going to show you that these have nothing to do with magnesium homeostasis. They do bring magnesium into cells, but it's not for that purpose. So one of the questions was, well, if we knock it out everywhere, could it be lethal because it affects transport through the placenta? So this SOX2 CRE doesn't express in visceral endoderm, and so would not interfere with the placenta. And this is a geo, so we can track it. So in all cases, all of the homozygous mice died. And then we tried to find out when they died, and they all die somewhere around we call it day 7.5. They die before day 7.5. That's unusual. No other trip is embryonic lethal in this way. So this geo allows us to track where it appears first and how it comes about in development. It first appears in heart, and then you can see it goes along the ventral surface, and eventually it spreads throughout and is present everywhere practically. This is a little artifact, this little notch here. But this TRPM7 is ubiquitous. Okay, so how do we study it? Well, we knock it out, it's embryonic lethal. So then we make CRE lines so that we can knock it out in specific cell types. And we did a lot of different cell types. It has major phenotypes wherever you knock it out. And so we decided, well, to get progress on this, let's knock it out in a cell that's most easy to study, and that's the immune system. I have to warn you, I hated immunology, always, and I never wanted to do CD4, CD8 kinds of studies, but the immunologists have powerful tools with their ability to count cells, and because of the tremendous number of markers and the fact that the tissue is already dissociated. So we chose lymphocytes to look at, and we made an LCK CRE knockout. This just shows that the current is knocked out. It's a CRE, and sometimes you measure like splenic cells by accident, so that's why you see some currents sometimes. But in cells that are the knockout, it's completely gone. So we first tested this magnesium influx idea. It's permeant to magnesium. It turns out that several channels, TRP channels are. Is it really doing anything in this case? And so this is TRPM7 knockout lymphocytes, and we're looking at magnesium influx. We had a hard time finding a good dye, but a group in Japan had recently made this dye called KMG 104AM. It's just right in the right KD for the range that we're looking in. And here we're looking at at this dye over time and changing magnesium. And you can see that wild type and knockouts, the magnesium influx doesn't differ from wild type. So on an acute time scale, the magnesium doesn't change. I wasn't surprised. None of us in the lab were surprised because the inward current is tiny from this channel, remember. But you could say, well, over time this will integrate. Even though the magnesium is small, it will change the amount of magnesium overall. But that's not true. We looked at inductively coupled plasma mass spec. This went to the geology department at Harvard, who has this. And looked at the total magnesium content in the knockout of the lymphocytes, and it doesn't vary. So there is no difference in total magnesium content in this tissue over time. So we would say that this has nothing to do with magnesium and nothing to do with magnesium regulation. And since then, we've been working on finding magnesium transporters, and we have found a couple of magnesium transporters which we think really do have something to do with magnesium homeostasis. So that leaves the question of what is that magnesium and calcium and zinc, which also permeates, what could it potentially be doing? And then what's the point of the kinase? So just to give you some feel for the problem that we've been struggling with is what kind of function does this kinase have? It's not easy to find the substrate of any kinase, the normal substrate of a kinase. It's easy to find potential substrates. Anyway, we looked at T cell maturation in thymus. T cells go through this process where they come from the bone marrow and they migrate into the cortex of the thymus, and then they get selected for. There's a positive selection and a negative selection, and you end up with single positive, either CD4 positive, MHC class 2 recognizing T cells, or single positive CD8 positive cells. And this negative selection, positive and negative selection, goes on in a kind of gradient from the cortex to the medulla. The thymic epithelia and the T cells interact with each other quite a lot in this process. The lymphocytes and the knockouts do survive, and they go to the intestine. These ones in brown are CD3 staining. You can see the T cells look just fine comparing the knockout and wild type. There is, in cell counts, there's a little lower numbers, but they look fine. And spleen, there's no difference between the knockout and the wild type. This is lymph node. They populate the lymph node fine. And in spleen, they also seem to be fine. They migrate out correctly and go to the right place and populate spleen. But if you look in the thymus, what you find is that the boundary between the medulla and the cortex disappears. So here we've shown it at a white line what that boundary is. This is just higher magnification of this. And this is the knockout. This boundary just can't be found in these 12-week-old mice. So the cortex and the medulla just sort of merge together. This is another example of that, showing wild type and knockout, that this boundary just disappears. And so we started doing cell counts, and we looked at T cell development. There's a partial developmental block in transition from double negative to double positive stage. There's a lot more cell counts I can show you, but I'm not going to go into all that. Basically, there's a failure of T cell maturation. And you can see it most clearly here in the CD25. CD25 is an IL-2 receptor, an interleukin receptor that is a marker on these T cells. You can see that in the knockouts, there's a failure to downregulate this high affinity interleukin-2 receptor. And this has consequences for how the T cell behaves. And if you look then at, with markers of medullary cells, these are with keratin type markers, look at cortical cells, you can see what happens. Here's the wild type and here's the knockout. And as you here's 4 weeks and 12 weeks. At 12 weeks, you can see that the medulla and the cortical boundaries are disappearing. So we wondered why that would be. We know it's complicated and no one has figured out the interaction between the cortex and the medulla. Or the thymocytes with the thymic epithelial cells. So we started looking. We knew that growth factors had a lot to do with this regulation, and we screened looking for up or down regulation of about 90 growth factors. And these were the main hits. And the interesting ones here, these go up and these go down in red. FGF7 and midkine activate STAT3. And STAT3 is known to maintain thymic medullary cells. Midkine induces this mesenchymal epithelial transition. And so what we think is happening is that many growth factors activate STAT3 by phosphorylation. STAT3 goes into the nucleus when it gets phosphorylated, and it controls many things: cell division, growth, apoptosis. People who have knocked out STAT3 know that these die about the same time as our TRPM7 knockouts. They die about before age 7. So the question then is, and before I get to that, we also then looked at STAT3. STAT3 is lost in medullary thymus epithelial cells. Here's, in the top is 4 weeks, and in the bottom is 12 weeks. You can see, compare this to this, there's just complete loss of this STAT3. Now, here's another example of that loss of STAT3. This is wild type, and here's the knockout. And you can see this is phosphorylated STAT3. That is STAT3 that goes to the nucleus, comparing it in wild type and knockout. So what we think happens is that somehow this kinase, the M7 kinase, phosphorylates something. It could phosphorylate STAT3 or could phosphorylate an intermediate, and that one step, at least for the T cell development, is missing in that case. So what we have is an ion channel now that has a kinase who has a big effect on early developmental events. And I'm not saying that it's all STAT3 dependent. In fact, we know it's not all STAT3 dependent. But that there are broad mechanisms controlled by this kinase, and we think that the channel itself somehow triggers the activation of this kinase. And so it is a system, this is what I believe is happening, it's a system whereby the small amount of calcium or magnesium or zinc entering is a way to trigger activation of the kinase only locally, and the kinase itself is doing the main business of this ion channel. So I'll stop there. This work involved all of these people, but especially a lot of work by Bimal Desai, who's an immunologist in the lab, and Jay Jenner did the knockouts. And I'll stop there.
Tom.
Hi. So I was curious if you have data or opinions or speculation about publications or ideas that this kinase may be a myosin-2 kinase that locally alters the peripheral cytoskeleton. I'm curious your thoughts on that.
Yeah, several people have asked me about it, and we've looked at it some. I can't say it doesn't phosphorylate any myosin, but I would say that still the central point of this is not picking a candidate and saying whether it phosphorylates it. The point is to find out what it phosphorylates normally. This is harder than it seems because we need to know what activates M7 normally before we can find that out, because we don't have a specific way of, let's say, normally activating M7 and then seeing what its substrates might be. Because it'll phosphorylate lots of things.
Yeah, on the first part of the TRPC5, you're proposing that it may release a small amount of calcium that aids perhaps in epithelial fusion.
Yeah.
Are there any inhibitors for TRPC5? Are there any inhibitors for TRPC5?
Yeah, the TRPs are pharmacology still in its infancy. There are TRPC5 inhibitors in pharmaceutical companies, but they're not available yet. There are kind of the best thing you can do to identify C5. It's unusual in that it's potentiated by lanthanum. Most calcium permeate channels are blocked by lanthanum. C5 is potentiated. There are other non-specific kinds of agents that affect it, but nothing very useful for pharmacologically.
Potentiated by lanthanum from the luminal side?
From the outside. Yeah, it binds to the turret of C5 and probably binds a calcium binding site.
Do you think there will be an easy explanation for ICANN as being mediated by one of these strip channels?
Sorry, I didn't hear.
Will there be ICANN, non-selective cation current,
Oh.
calcium activated turn out to be one of these trip channels, do you think?
Oh, yeah, certainly. If you mean, for example, the 25 picosemen channel discovered long ago in Ayer's lab, that is known now. That's TRPM4B, and it's sometimes called ICANN, if that's what you mean by ICANN. Yeah, that's a calcium-activated 25 picosemen channel. That's TRPM4B.
Did you mention that there are trafficking defects in TRPM mutants?
Yeah. So in TRPM-L3, we've studied most TRPM-L3 knockout mouse. TRPM-L3 is a gain of function in varitant Wadler mice. Those mice have gain of function where ML3 is brought to the membrane. It turns out it activates that channel, and it allows that channel to be brought to the surface. Actually, I think this is the strongest evidence that they play a role in vesicular fusion. And that causes calcium overload and cell death in melanocytes and in hearing cells and so forth. If you do knockdowns of mucolipins, 1, 2, or 3, you find, for example, very large endosomes formed, disruptions in, and depending on which one you're knocking out and which tissue, but you find big disruptions in lysosomes and endosomes. Yeah, lots of different effects there.
Any diseases associated with the mutations or overexpression of these channels? Any diseases?
Any diseases?
Yeah.
Well, by overexpression or by any. Well, there's lots of diseases. So polycystic kidney disease, the most common genetic disease in the country, but has a late onset, usually in the 50s, is a mutation of TRPP2, that is PKD2, and PKD1, both of which are in the TRP family. So that very common renal failure is through PKDs. Mucolipidosis is a TRP channel disease, and there's recently been discovered a bone density disease, high bone density, discovered a very rare disease caused by a TRP mutation. And there's several others, but I've got a whole list of them, but none of the others are as prominent as the ones I've just mentioned.
Any other questions? If not, thank you. Okay.
Dr. Regan has been a leader in this area of protein design almost since the field existed. She got her bachelor's degree from Oxford University, then came to the United States to get her Ph.D. at MIT with Paul Schimmel of Cantor and Schimmel fame. She went on to do her postdoc with Bill DeGrotto, who was then in the research arm of DuPont, where they published the first real success stories of designing novel proteins, mainly helical bundles. She then moved on to the Department of Molecular Biophysics and Biochemistry at Yale, where she's variously worked on designing novel metal binding sites into proteins, testing the extent of protein plasticity. In one case, she was able to convert an all-beta sheet to an all-alpha helical protein, mutating a surprisingly small number of positions. She's since moved on to looking at molecular recognition and applying some of these protein design principles to potential therapeutic applications. And so, welcome.
Okay. Well, I'm very pleased to be here. I'm going to talk to you about my design work on repeat proteins, and I put a few here that you may have come across: ankyrin, heat repeats, leucine-rich repeats. The ones I'm going to be talking about are TPR repeats. Now, they're not TRP repeats or TRP repeats that we heard in the last talk, though I did get email when I first started publishing from someone who worked on those asking if I could advise, and I said, well, no. It stands for 34 amino acids. The reason I started working on this class of protein is because I thought it would be a simplification of both the protein folding problem and design for molecular recognition. And if you just take a look at these schematics, you can get a hint of why that is. If you look at in each case, one repeat is colored differently from the rest. And if you think about this structure versus that of a globular protein, virtually any protein you can think of, then they would have many globular proteins, many long-range interactions. Residue 10 would likely contact residue 100 and so on. But here we have interactions within a repeat and between this repeat and its neighbor. And this repeat does not interact with repeats, 2 away or 3 away. So just kind of conceptually, that simplifies the protein folding issue. And I like to think of it in this way, molecular Lego. So now we're going to talk exclusively about these TPR repeats, though I would think many of the things I say about their structure and stability would apply to any class of repeats. It's just that these are proven very versatile, and so they've become our model system of choice. So if you think of a single brick as being one repeat, which is helix turn helix, then to create proteins, you have to just stack the bricks. And this is the analogy is that the interfaces between the bricks can be constant, so you can introduce different colored bricks, different functionalities onto the different repeats, but still just put them together in this relatively simple fashion to make a protein. And so one of the first things we did was to design the repeat, the brick, and we designed what I would call a consensus repeat. So we just aligned all the sequences, thousands of sequences, and just looked for the characteristic features that made these TPRs as opposed to 4-helix bundles, as opposed to myoglobin. And you see a specific pattern of hydrophobic residues in this little picture, just going along the linear sequence, and the red bars are the conserved positions. And so we make a protein out of those residues with virtually anything at these other positions. And here's a crystal structure of it. And it's an interesting that it's very rich in hydrophobic residues, and the hydrophobic aromatic residues, and they pack against small residues like this. And so if you actually, I don't have it on this picture, but you would see that there's a conservation. This trip matches up with this alanine and so on. So it's kind of a nice kind of regular structure, and we can make this our idealized version. So now we can put those bricks together. Is it true that we can just stack up those TPRs? Well, we can. And these are crystal structures of an 8. And I'm showing you this view of an 8 because 8 makes one complete circle of the turn, rather, of the superhelix. And this is 20, just showing you what that looks like. And you can, and remember, one repeat is 2 helices. And interestingly, for some completely different reason to do with protein folding studies that I won't be talking about, we were doing single molecule fluorescence measurements just to look at how these things behave. And basically, you measure their diffusion, and from that, calculate hydrodynamic radius. And if we take a structure from the crystal and calculate hydrodynamic radius and plot that versus our measured value, we kind of see this straight line. In other words, over this range from about 4 to 20, they are behaving like stiff rods. Because you might wonder, I didn't know what the persistence length of a solenoid would be. Would there be any floppiness or so on? And I mention this because for design purposes, it's nice to know where everything is going to be. And that just because we have a crystal structure, it's nice to know that in solution, at least over this length, we know where binding sites are going to be positioned. And you'll see in a moment why that's important. The other thing that we're able to do, again, I think this is made easier because of the repeat nature of the proteins, is we can modulate their stability over a tremendous range. So if you just look at the top 2 panels here, all we're doing is we're doing thermal melts, temperature versus how much is unfolded. And this top region, it's the same length. So you do a lot of things with this we call C for consensus, TPR3, 3 repeats, because that 3 repeat is the smallest functional size. And we're going to be starting to make things that actually bind ligands. So here's a natural one, and here's one of our designed ones. You see we can make them much, much more stable than the natural one. And also we can play about even more with stability. So here we've got one that melts about 40, 80, and then this guy doesn't really melt until over 100. So they're very kind of robust and engineerable. And also, as we increase the number of repeats, so now we have the same repeat, we're just increasing from 3, 4, 5, all the way up, then the stability changes in a predictable fashion. So you might say intuitively, well, of course, I'd expect them to be more stable as they get longer. But in fact, you can describe it much more than just intuitively, because if you use what we would call an Ising model from physics, or what I would call a Zim-Bragg from protein work, in which you say, okay, every unit is either folded or unfolded. There's an energy associated with that. And a folded unit can be either next to another folded unit, so it's an interaction energy, or it cannot. So there's just two energies and the number of repeats. And you can predict any protein in this set. So in fact, when we were doing this, we were kind of making them and measuring them. And we predicted, okay, we've made 5 and 6, what's 20 going to look like? And the data falls exactly on our prediction, which is not the case for globular proteins. Usually, if you're making a mutation in a globular protein, you can make an educated guess, but probably a 50% chance of it being stabilizing or destabilizing. So this level of control was really quite pleasing to us, that we knew what was going to happen if we made these things. So that was sort of the background to tell you that we have quite a lot of control over the structure and stability. But what I'd really like to talk more about here is making these modules bind different things, and different things that might be useful for investigating biological systems or for diagnostics or whatever. So we have to just talk a little bit now about ligand binding. So a natural system that uses TPRs is the HSP70, HSP90 folding system in eukaryotic cells. And I'll get back to this in a moment. For the present, all we need to know is that there is one of these 3 TPR units that binds this peptide at the C-terminus of HSP70, and there's a different one that binds the peptide at the C-terminus of 90. And actually, these two, TPR1 with peptide, TPR2A with peptide, were the first co-crystal structures done by Schufler and colleagues. And you see that the peptide is bound on an extended conformation. And if you look down, you see there's kind of a concave little surface where it binds and a convex surface. Now, if you look at all the TPR structures that are available, either with or without ligand bound, and superimpose them, there's virtually no backbone movement upon ligand binding. There's definitely not a systematic closing, as you might have thought, nothing like that. And that's important because if we want to model conformations of side chains or something like that, then, or dock a sequence into a TPR, it's much easier if we don't have to worry about backbone movement. And these are not. So that's good. That's helpful. The peptide is typically bound. It's 5 to 7 amino acids. It's typically bound in an extended conformation like this, which again, kind of thinking ahead to what we're going to be doing, if we want to make TPRs that bind any peptide you'd like, then it would be preferable to be recognized in extended versus, say, some motifs that recognize polyproline 2 or an alpha helix. This is more versatile. And finally, the residues involved in binding are readily identified. And I just want to take a moment to tell you what I mean by that. So in the course of studying TPRs, then we were doing a bit more detailed analysis of sequences than just, oh, these are conserved, these are hydrophobic, these are specifying the fold. And we were calculating in each position the degree of conservation. So again, we've got our list of about 1,000 sequences, and we're just calculating color-coded from red to blue, how variable they are. And what we're doing in this figure is mapping those variabilities onto the co-crystal structure of the 2A and 90 peptide. If you look first over here, so all the red conserved residues are buried, as you might expect, right? They're hydrophobic residues specifying the fold. On the back face, you're pretty variable. But on the front face, this was the surprise to us, sort of around where the peptide binds, you're actually hypervariable. So when my postdoc, Tom McGleary, who did this, showed me this picture, I said, oh my goodness, it's just like Wu and Cabot and antibodies. It's just like this, when way back in the 70s, they looked at the sequences of different antibodies, and they defined the complementarity determining regions because they were the most variable amino acids. And then this all came true when people mapped binding sites on antigens. And so when I saw this, it said to me that, well, it's just the same, of course. And then a few minutes later, well, how could we have missed this? Of course, if we've got a constant framework and we're using it in the cell to bind lots of different peptides, then of course we'd expect this. Then we went on to show that actually for some other scaffold proteins, like anchorins and I forget what the others were we did, that you see this. So just to kind of explain why this was interesting to us, typically people will look at sequences and say, okay, a conserved hydrophobic residues, that specifies the fold, they'll be buried. And I would say that too. And then they'll look and say, oh, surface residues that are conserved, they must be important. They must be the binding site. And that's often true if you're just binding one ligand. But if you've got a motif that binds a lot of different ligands, that's not true. And when we figured this out, we were surprised that no one had noticed that before, but in fact, they hadn't. But that's what I mean by it's easy. So these, it will come up again that, so we were showing those even in the absence of a crystal structure. We're just mapping them onto a crystal structure. We can do it from sequences alone, and we've done it for others too. So therefore, you might think, well, conversely, if at those blue positions, you've got a subset of TPRs that have the identical residues at those positions, that suggests that they must bind the same ligand, just this kind of very simple hypothesis for recognition. So if we take one of our designed TPRs and we graft onto those the recognition residues for the TPR2A here, then we should now make our designed scaffold into a peptide binding module. And in fact, that works. So here, this is just using a Bierker experiment. This is increasing amount of peptide and nothing, no binding to the parent molecule. But when we graft on these residues, we see binding. This is a crystal structure of the designed TPR with the peptide bound. And actually, the binding is a little less tight than the natural pair in this case. And if you, I won't go into this, but if you look in detail, then there are some interactions that are not, most of them are well reproduced, but there are a couple that aren't, and I think we lose a bit of binding affinity. But I think that kind of for a proof of principle, to kind of emphasize the modularity of recognition, then we were pretty excited by this. Also, we showed that because these molecules are so stable, they're really tolerant of changes in charge. And so what we're doing here is we're looking at the back face, so not the face where the peptide binds, but the back face. And we're seeing what happens because if we're trying to bind this peptide, it's very negatively charged. What happens to affinity if we change that back face from completely negative to neutral to completely positive? And the protein stays happy throughout all this. This one melts at over 100 degrees, and these two in the 80s. So we've got quite a lot to play with there. And you'll see that if we make this negatively charged to bind a negatively charged ligand, we don't do very well with the binding. But then the neutral one, and now the positively charged one, binds very well indeed. And the key experiment to go with that is to show it's still specific. Because after you've been excited for a moment by that, then you think, well, it's a big blue positive blob of binding a negative peptide. Why are you so excited and surprised? And I was excited and kept being excited because this is the cognate peptide that we were intending to bind, MEEVD. And this is a related, in fact, the C-terminus of HSP70, which is equally negatively charged. But you can see that it doesn't bind that at all, and it binds tightly to this. And in fact, what we've made, which goes under the name of CTPR3 90+, it actually binds more tightly and with greater discrimination against the non-cognate than does the natural TPR2A. And I'll show you this is useful for dissecting the mechanism of HSP90-mediated interactions and so on. We'll come back to that.
Does it bind in the same way?
Yes, yes.
It still binds in the front face, even though the back face?
Oh, yes. So actually in this, yes. Because we did HS at QC NMR shift mapping to show that. Yes. Yeah, no, we did worry about that. It hasn't just popped around the back. I think if it had popped around the back, we wouldn't have the specificity. So, okay. So let's just go back to HSP90-mediated protein folding. So there are a subset of proteins, and many just somewhat coincidentally are associated with cancer. So HER2, cell surface breast cancer protein, ACT, kinase that you heard about earlier. And there seems to be no kind of structural similarities between these proteins, but they all seem to be dependent upon 90 for folding. So what do I mean by that? I mean that this set of proteins, here is unfolded, starts folding on HSP70 and then gets passed to HSP90 where folding is completed. And if this is, for some reason, doesn't happen, then that protein is degraded by a TPR ubiquitin ligase called CHIP. And because the HSP90 dependent proteins are associated with cancer, then inhibition of HSP90 is an active target, and there are some molecules in phase 1 and phase 2 trials. And I'll tell you about those in a moment. So there's a class of molecules called geldanomycin or 17-AAG. And they were initially found because in general screens at the NCI, they killed cancer cells. And the first thought was that they actually inhibited kinases because kinase levels went down. But it was later found that the kinase levels went down because the kinases weren't folded properly. It wasn't a direct inhibition of the kinases. And so here, you can't see it very well, but the 17-AAG binds to the N-terminal ATP binding site of 90. So here's a little schematic of 90 that tells you virtually nothing, except here's the N-terminal ATP binding site. That's the active site where geldanomycin binds. Here's the rest of it, and here's the little peptide that makes it part of that complex with 70. And just to show you the effectiveness of inhibiting 90, here are HER2-positive breast cancer cells, BT474, I think, and here is control, Western blot, HER2 levels. It's a HER2-positive cell line, so you're overproducing HER2. If you treat with 17-AAG, you inhibit or actually push towards degradation. And in a xenograft mouse model of human breast cancer, then treatment with 17-AAG reduces the cancer size. So this is, at some level, a validation that inhibition of 90 is a route to sort of general anti-cancer agents. There are a few problems with 17-AAG and its relatives. It's quite liver toxic, it's low solubility, and it has this unexpected and poorly understood phenomenon of inducing HSP70. So if you look here, here is just untreated versus treated. For reasons that are not well understood, you get this massive increase in 70. Now, 70 has got some level of anti-apoptotic activity, so this isn't the best thing to be doing if you want to kill the cells. There is actually a whole group of researchers who use 17-AAG just for this purpose, because inducing 70 like this solubilizes glutamine-rich aggregates to some extent, so there's a whole field of study. So do you have a question? No. So what we thought was, well, if 90 has to be in this complex to function to get the protein passed from 70 to 90, how about we inhibit the interaction of HOP with 90? And there was data from Mike Chinkers, who a long time ago had shown that if you overexpress the TPR domain of PP5, you inhibited glucocorticoid receptor maturation, which is a client protein of 90. So our first thought, well, what about our designed CTPR protein that binds to this tail more tightly and more specifically? If we were to introduce that into breast cancer cells, could we have what I'd call a dominant negative effect of this module now outcompeting and preventing 90 forming this functional complex? Oh, yeah. And in fact, that's what happens. So here we have different amounts of that protein introduced into the cell, and we're following HER2 and phosphorylated HER2. And you can see we get a real diminution of the levels of the active form. We don't get an induction of 70, which is good, and the cells are killed, which is what happens if you inhibit 90 with 17-AAG. So this is one example. Of course, you're not going to use this as a therapeutic, but it kind of just shows you that modules that are designed and introduced into cells can actually have activity and can be useful in understanding mechanisms. But if you wanted to think about actually going this route to drugs, then you're not going to be dosing someone with these peptides, or these proteins rather. And so I'm just going to go over the details of this briefly, but we set up an assay where we look for small molecule inhibitors of the TPR-HSP90 peptide interaction. And I should say that because it's a relatively small area, the odds are in your favor, as opposed to some complex that's got a massive surface area buried, that you might find something. So we were looking at small molecule inhibitors with different libraries. And this is just to say that we went through and did a lot of controls. And just to come down here, at the end, we had a set of proteins that were active in 2 different in vitro assays, could be detected binding to 2A by ITC. And most importantly, they killed cells, and by Western blots, they inhibited HER2 levels. And in quite a dense form, this data is shown here. I just want to point out a few things, not everything. So when we identified this class of compounds, then the NCI who was doing this particular screen bought any commercially available relatives, and we found that 5 out of the 6 were active in vitro, but 1 wasn't. And these are all showing no activity in a control. And then in cell killing, we found that this guy who was inactive in vitro was also inactive in cell killing, whereas the other guys were active. And we get a reduction in HER2 levels. So what this little story is to tell you was that by designing the protein, we had this sort of proof of principle experiment that we could inhibit a protein-protein interaction and then take that forward. And this is obviously still in progress to get now small molecules that do the same thing, that obviously have the potential for further development. I want to go back now to show you some different designs for TPRs. So that was fine, but remember, we're just making a TPR that's got the same specificity as a TPR we already know. We're not putting a new specificity into it. We're just sort of making it better than the one. And you can think of these, and if anyone has a use of these that they think would be appropriate, you can come and talk to me afterwards, because we are interested in what we might use them for. But the idea is that if we can make these TPR modules or T modules to bind any desired sequence, then when you have a c-Myc tag or something on a protein, you could actually use one of these rather than the huge IgG to bind it. And also, they are functional in the cell and so on. So I just want to show you a couple of examples of making these modules with different specificities. So the first one was sort of a designed pair. So we kept certain of the interactions from this known CTPR 390 HSP90 peptide, but in this region here, we substituted a charged residue at the C-terminus for a hydrophobic residue. And then we made this little pocket of hydrophobic residues on the TPR. So basically, we have this very small library where we have these different hydrophobics at those positions, and then we have a library of peptides, and we can look to see what the binding is of these, and what combinations, if any, on the TPR give us a binding pocket now for hydrophobics. And so we found that many of these work. One that we like best is this MMY, so methionine tyrosine, which binds the MEF peptide. And this is just some ITC and BIACOR data on that. And I kind of like this. This is my favorite experiment, where we look at the binding energy of peptides with different hydrophobic substitutions at the C-terminus, and we plot that versus the hypothetical surface area that hydrophobic residue would bury if it went into this pocket. And we kind of see this linear correlation with free energy per unit surface area that's very consistent with measures people have made when they play around with the hydrophobic core of a protein. The other thing is, you think, well, hey, so the bigger you get, the better. So if Phi is good, is tryptophan better? Well, no, we've reached the limit with Phi, because tryptophan is too big to fit in this, and you don't get any extra energy if you go to tryptophan, whereas in this series you do. So now we've got a new peptide tag, and you can use them. I'll just show you a couple of things, not everything, in Western blots. So here, this is sort of just a first experiment where we've tagged with MEVD or MEVF, and we have the TPR with a his tag, and we have secondary antibodies. This is just to look at the first stage. We're not saying this is going to be a useful method itself, but we can pick up the correct tagged GST with this peptide, with this TPR, and we don't pick up the wrongly tagged one and not alone. But more, I think more usefully than that is because we're making this protein in E. coli, we can directly tag it with, well, whatever we like, but in this case with GFP. And so now you can do essentially a one-step Western blot where you transfer, here's just dot blot titration, you transfer and block and then probe with the TPR fusion, and you can do detection on, this wasn't trying to optimize, this was just on our DNA light box. So I think that there's a lot of potential for actually making things that you can use. And then the last thing I want to say, talk about is, well, okay, so far we've made a TPR that binds a natural peptide, and we've twiddled around a little bit with recognition specificity, with useful consequences. But how about selecting for TPRs? How about if we want a completely different sequence to be bound by the TPR? And I was keen not to do this on just some random peptide that we thought of. I wanted to do it on something that there was a biological reason that we were interested in that protein. And I was at Sloan Kettering, and Nicola Pavlitic's student was giving a talk about this crystal structure she'd solved, which is several years ago now. And it's the BRCA2 protein, which is in this gray color, bound to DNA. And it has this small 70 amino acid DSS1 peptide. And the big deal was that she couldn't get expression and she couldn't get crystals unless she co-expressed this little guy. It was kind of central. And then a group mutated this in Ustalago and showed that there were changes in DNA repair by BRCA2. So it was very important for BRCA2, and I thought, well, it was an interesting protein to track, you know, when it became associated and so on. And then the other thing that I thought was interesting was yeast has a DSS1 equivalent called SEM1, but having told you all this about BRCA2, yeast doesn't have a BRCA2. And so I thought, what the heck's it doing in yeast? So it's kind of an interesting thing. And it's small because we thought, well, maybe we can bind to it and inhibit its activity in that way I just showed you for HSP90. Or, and this is a more general thing, track location, because if it's too, I think it is, it's too small to tag directly with GFP without inhibiting function. So you can imagine maybe expressing the GFP tagged TPRs in the cell and just lighting it up at the times you want. That was kind of the idea. So that's all very well, but we didn't approach this with trying to do any kind of design or docking to start with. We just said, let's make a library and screen. And so remember in that slide where I said, these are the blue residues, they're hypervariable. Well, those were, of course, the ones we wanted to make libraries at those positions. And we, just because we were doing this in E. coli, we wanted our library size to be no bigger than about 10 to the 9. So we had to, most of these were objective choices, but we had to miss a couple out because our library had been too big that if we'd have just looked at that hypervariability, we might have put them in. So these positions shown in blue, we made libraries where we had all 19 different amino acids. We didn't have cysteine, and we did this by triplet codon mutagenesis. So we had equal representation of all 19 amino acids each position. So that's the library. And then, of course, we need to be able to screen that library. And if you look at the top here first, for other reasons, Neil Ghosh in my lab had been playing around with green fluorescent protein. And he had found that if we split GFP into two halves, an N-terminal half and a C-terminal half, they're not fluorescent. And if you express those in the cell at the same time, they don't reassemble and become fluorescent. But if you tag them with two interacting molecules, then the interaction of these two brings these into proximity, and you kind of fold in a fluorescent. And he did this first with leucine zippers, and I was keen to test it with a TPR system. And indeed, the thing I liked about it, because you might think, well, why don't you screen the library in a different way? Why don't you do phage display, yeast to hybrid, whatever? I liked the fact that It was only the cognate pair that gave us green, and then this TPR with different peptides didn't, or this TPR with a different peptide didn't. So I like the idea that I thought we were not likely to get false positives. We might miss some, but we might get false negatives. But I thought we were going to get specificity of binding. So what we did was we had our TPR library on one half and our target DSS1 on the other. And then we used FAGs, cell sorting, sorted, grew, and then sequenced. And I was very pleased when we saw this. We see conservation of negatively charged residue, hydrophobic. And if you look where we are on the peptide, on the TPR rather, we have this stripe down that helix. So in this selection, not much conservation here. There might be, we didn't try too hard to find it, but this clearly just jumps out at you that you've got conservation down that helix, which is a sign that you've selected for something. I mean, it might not be what you want, but you've selected for something. Well, that last one that just went past was just, yes, we can make the TPRs and show by ITC and Biacore that they do bind. And what then we wanted to do was, this was what we were selecting against. And we wanted to map the peptide binding site. And remember, we expect the peptide to be binding in extended conformation. And so we made this half, and the middle. And surprisingly, none of those bound. And I was a little bit worried at that point because, well, there was only one possible peptide sequence of 5 to 7 amino acids that could be the recognition that we weren't covering here. And that seemed a little bit too unlikely to me, that was coincidence we were missing it. And I started thinking, well, maybe this is a beta hairpin or something. But actually what Spurcotarena, my postdoc, found was that the peptide is helical. So the peptide alone actually forms helix, and then in complex with the TPR, it seems also to be helical. And so then I think the more convincing data is shown here. So then we said, well, if it's helical, that completely changes how we would dock it against the TPR. And there was really only one orientation that you could satisfy this negatively charged hydrophobic, which was with this arginine as the positively charged hydrophobic, and we're not quite sure which hydrophobic over here. And so what ITSPA did was she made some peptides, some variants of DSS1 in which she mutated residues. So if you mutate arginine 57, you diminish binding, but you don't abolish it. If you mutate leucine 60, you kind of abolish binding. And so we kind of like that. We think we're trying to grow crystals a bit, but we think that this is likely the orientation. And the thing that I want to emphasize is this library was selected from a completely randomized, non-tailored library. You know, we weren't trying, this library was not intended to just be a DSS1 binder. So you can imagine that additional features can be designed or selected onto this. Now we've got a starting point. Also, for other peptides that you're interested in, you can kind of fine-tune the library so it's not completely randomized. But the last thing I want to show you is the effect of expressing this TPR in yeast. Because remember, part of the point of doing TPRs rather than antibodies was that we'd be able to use them in vivo, either to bind up and inhibit or to tag. And so we decided to do this in yeast because just because it was easier to work with yeast, basically. And the SEM1 equivalent of DSS1 also bound to our TPR. And so let me just show you that data. So wild-type yeast grows well at 25 and 37 degrees. Yeast with a chromosomal knockout of SEM1 still grows fine at 25, but it's dead at 37. If you complement the yeast deletion with human DSS1, you recover growth. And so now what we did was to express our TPRs that bind both the endogenous SEM1, or they would also bind DSS1. So here, this is wild-type yeast expressing the TPR, grows at this temperature, but not at the high temperature. And we think what's happening here is the TPR is binding up the endogenous SEM1 and inhibiting activity. This, of course, is dead because this is just the deletion. Here, deletion of SEM1 with DSS1 on a plasmid, so the levels are a bit higher than the endogenous SEM1 would be here. And now we get a little bit of growth, but then none. And I think that's because there's more of the DSS1 here. And so we were pleased to see that these are functional in vivo. And actually, if you think of that slide I showed you, then we don't really know. There are now like 3 different things that are being proposed in yeast, what SEM1 might bind to and why it's important. But these would be tools to address what the SEM1/DSS1 is doing in yeast. So what interaction has been disrupted here is its interaction with the proteasome, which has been suggested, its interaction with RNA processing, because now we have a tool by which to investigate. And so I'm going to finish up here just trying to remind you what we talked about. I started off telling you about how we can understand and manipulate the structure and stability of TPR modules and arrays. And this was the work of Aitzber, Cortorena, Tommy Kayanda, and Tom McGleary, who's now at Ohio State in the chemistry department. And then I showed you how we can understand and manipulate and create TPRs with new activities. And this was work of Tina and Aitzber and Meredith. And then I showed you that, in fact, we can kind of take data we have by using TPRs, and there are cases where we can use small molecules, and that was Fong Yi's work. And that's all. Thank you.
Any questions? Are TPRs ever naturally modified in a post-translational way to alter their binding?
I don't think anyone's looked. I would doubt it, actually, but I don't think anyone's systematically looked for that. I say I would doubt it because often they can be expressed in E. coli and retain the same binding specificity.
Is there any known reason why the yeast at 25 is happy and the 57 responds to this?
Oh, well, right, so because they don't know exactly what DSS1 is doing, it depends what you think the target of DSS1 is. If you think the target of DSS1 is the proteasome, and that you need DSS1, sorry, SEM1 to be active proteasome to get rid of proteins that are not doing too well at high temperature. And so if you do, and so it's necessary for activity of the proteasome. And so if you don't have an active protease, that's one. Though, as I sort of alluded to, there are other recent suggestions that SEM1's involved in RNA processing or RNA export. So it's not, yeah, that whatever it's doing is needed at 37, be that the proteasome activity or the other.
Do the small molecule hits mimic the structure of the peptide in the extended conformation, and do they bind in a similar way to the TPR?
Well, wouldn't we like to know that? So the 2 assays we have, the so-called alpha screen bead assay and an anisotropy assay, they're dependent upon inhibiting an interaction. So all our point mutations in the TPR, we can't look at them because we have to have the binding to start with. We can do ITC to look at the direct interaction, but the binding is quite weak, so I don't think that mapping our mutants will help there. Basically, we're trying to crystallize it. We have small crystals. I think that's the way to go. We're also doing a bit of that HSQC mapping to see that. I think it must be, you can't dock it, so you can't dock it at a unique place. Yeah. There are like 7 places.
There's no groove, really.
Well, I think it's partly because the molecule is small, and though there is a groove, there's this. I mean, if you were trying to dock, but even if you say you're going to dock it in there, you can't get a unique place that's just.
Do you see some generalities about in the different hits? Are they similar to each other?
Yes, I should have said that. So the ones that make it all the way through all the negative controls, then they're a family of things that have got just different ring substitutions. And there's one position that the one that's inactive is, there's only kind of 2 places that they're substituted. One of them, it can tolerate sort of anything. The other, you have to be methyl or larger, so the proton at that position doesn't work. So yeah, I should have said that. Some of the other hits along the way that didn't make it all the way through weren't related, but then they didn't make it all the way through to the in vivo assays and so on.
There are no more questions on this.